Last week we completed the topic on nucleic acid metabolism. That's just a brief one to take in 2 lectures. We first took the catabolism, and then in the last lecture we took the anabolism of nucleic acids. Today we're continuing with the enzymes and looking at enzyme action mechanisms. I think in first year we did something on enzymes. I think when we were doing something on proteins, we touched briefly on the function of proteins as enzymes. And so we did look at naming, classification, and some basics of enzymology. We did look at that. So some of those things you have to go back and revise them, because you already have those materials, so we don't have to be repeating them. So that's maybe what we have on enzymes too. We can take it in 2 or 3 lectures. And then the second half of the course, which is going to be on biomembranes, we'll move on to that and pay more attention to the biomembranes. So today we take enzyme action mechanisms, which these things are quite straightforward. So we don't take so much of a time, maybe in an hour plus, maybe 1 hour, 20 minutes, 30 minutes. I should finish talking, and then if you have any questions, if you have any contributions, you can make. As we always do, the chat is on. So if you have any questions along the lecture, you can type in the chat and I'll respond to it. Or you can note your questions or your contributions down at the end. I will give the floor. If you wish to speak, you can raise your hand. I will enable your microphone, call your name, and then you speak. Then I respond. All right, so quickly, I want to draw your attention to something which we did already in the first year, which is on factors affecting enzyme activity. That you should, in general, the things we did, you should go and revise them, but we can go over this quickly. So we took some 4 main factors which affect enzyme activity, which are enzyme concentration, substrate concentration, temperature, and pH. And how do these affect enzyme activity? So when you look at the relationship between enzyme concentration and the rate of the chemical reaction, a biochemical reaction that the enzyme will be catalyzing, you notice a linear relationship. And that is to say that with increasing concentration of enzyme, you have an increased rate of reaction. And then that is if you have all other factors constant, I mean the factors which we have already seen, like substrate concentration, temperature, and pH. And so you see such a linear relationship with enzyme concentration and the rate of reaction. It's logical that you have enzyme molecules, which are proteins that will be acting on substrate and converting that substrate into a product, so that the more enzymes you have, the more the activity of the enzyme on the substrate. And so the faster the conversion of that substrate into the product. How about substrate concentration? You notice that for substrate concentration, there is an initial linear relationship where there is an increase in the rate of the chemical reaction with increase in substrate concentration. But you notice that at a point, this linear relationship flattens out so that the rate of the chemical reaction becomes constant from a certain concentration of substrate. And bear in mind that you have enzyme concentration being constant with the other factors. Now, to understand that, if you have substrates, let's say we have the substrates which are being represented with, these red spheres, and you have the enzyme being represented with these bean-like shapes, so that you have the active center, center where the substrate is going to bind onto the enzyme for the enzyme to catalyze a reaction on that substrate, being shown as that insertion in that bean-like shape. Now, in the beginning, where you have a low substrate, so you can see that you have the amount of enzymes present being more than the amount of substrate present. In this case, the enzymes will quickly act on the substrate, so that, as you can see, you still have more enzymes that are free in the reaction mixture. So if you add more substrate into that reaction mixture, those substrates have free enzymes that can act on them. And so you will notice a linear relationship, that is an increase in the rate of reaction when in the beginning you add more substrate in that mixture, because there will be free enzyme molecules to act on the extra substrate molecules which has been added into the mixture. But you see, in the last end, when further addition of substrates is done, you have now a saturated reaction mixture where you have more substrates than enzyme molecules. In this instance, what you notice is that all the enzyme molecules are occupied. They are all engaged with substrate molecules. What it means is that even if you continue to add more substrate in this mixture, the enzyme molecules are already occupied. They are all engaged. And so there will not be any change in the rate of the reaction. The amount of product which is going to be formed at any point in time will not change. It will be constant, which is why you see the flattening of the curve. See that the rate of the reaction becomes constant at that point, like you have reached the maximum rate of reaction, and so there will not be any change even when there is increase in substrate concentration. How about temperature? Now you notice that with temperature, you have an optimal temperature. So this curve is for the general physiological temperature of, say, human beings, for instance, where we have our physiological temperature around 36.6, approximately 37 degrees Celsius. Now, it is at this temperature that you have the enzymes in our bodies being optimally active. So you can see the highest rate of reaction is around that, 36, 37 degrees Celsius. Now, when temperature increases, what happens? Why do we see that the rate of reaction reduces with increase of temperature? You remember that we did see that enzymes were mostly proteins, and some enzymes are RNAs, some catalytic RNAs, which are involved in nucleic acid processing. Now, these things we are talking about, proteins and nucleic acids, you know them to be biomolecules. And these biomolecules at high temperatures, what happens to them? They get denatured. They lose their configuration. Their functional configurations are lost. And so they can no longer be functional as enzymes when you have temperature increasing, particularly when you have temperature increasing beyond 40 degrees Celsius. I mean, this explains why it's a fever that results into increase in temperature, like body temperature, can be serious, because what it means is that the proteins in the body, not only enzymes, but proteins or maybe even things like hemoglobin, red blood cells, will begin to break down with high temperatures. And with such high body temperatures, what will happen if you are having breakdown of these proteins, like the red blood cell, say the hemoglobin in red blood cells, for instance, or even the uh, um, nucleic acids, say RNAs, which are also, carrying out several functions, or you have some globulins, proteins, which are acting as some immune cells. If all these things are breaking down, then of course you are going to have a situation of some anemia, or it can rapidly result into not enough oxygen being carried to the tissues, and that can result into some serious cellular suffocation, and it can become fatal. And so you see that when body temperature increases in a patient, for instance, it becomes an emergency situation for health practitioners to deal with very quickly. How about lower temperatures than that optimal temperature? You also notice that the lower temperatures also result in decrease in the rate of reaction. Now, this you can also look at it with a practical application of the process of refrigeration and freezing in food preservation. What is food spoilage? The major aspect of food spoilage is about microorganisms that grow, feed on food, and release byproducts, which are toxins, into the food substance, making the food unwholesome for human consumption, so that when you consume such a food, you get food poisoning. Now, how does refrigeration or freezing help with food preservation? Now, generally, if you take food substances that contain, say, about 70% plus water content, such food, is a good environment for bacterial growth. And so you will see that food substances like maybe fruit juice, milk, when you buy them, even though there is an expiry date that is written on them, there is also an instruction that when you open them, you should consume them within a certain period of time, like say maybe 24 hours or sometimes even less. And that is because once you open the seal, the food is now exposed to oxygen. And that will speed up the bacterial growth in such food. So what it means is that the fact that you are refrigerating some food or freezing the food doesn't mean that the food will not spoil. The food can still get spoiled, even under such condition. The only thing is that refrigeration is reducing the rate of reaction. What reaction? Biochemical reactions that the enzymes of the bacteria are catalyzing. Because the bacteria is feeding on the food substance, like say maybe if it is some lactobacillus, for instance, that's, bacteria is feeding on the milk sugar lactose. Now, if it feeds on the sugar, it breaks it down into some aldehyde, acetaldehyde, acid, and all of these will give some sour taste, bad odor over time to the milk. It make it unwholesome. For instance, or you take maybe bread, which has some amount of moisture in it. So if you have moisture in food, which is about maybe 20% plus thereabout, that food environment is also conducive for fungal growth. And so what you see is that the reduction of temperature results in the decrease of enzymatic reactions in these microorganisms. And so that can be applied in food preservation. It is just to slow down the process of food spoilage. It doesn't stop it completely, but it will slow it down so that the food can be preserved for a longer time. If the food is kept in the fridge or freezer for too long a time, the food can still get spoiled because the reaction is still ongoing, except that is going on at a very slow rate. But of course, you do have this one I'm talking about, say organisms which are, having this optimal temperature, say around 37 degrees Celsius, which is like, this bacteria, fungi. But of course, you do have some thermophilic bacteria that live in conditions that are of much, much higher temperatures, like temperatures over 100 degrees Celsius. So, for instance, you have some bacteria in deep sea where, for instance, petroleum is being drilled. Let's say like Takrady on the FPSO. Deep down, there are some bacteria in deep sea that can withstand temperatures of even over 100 degrees Celsius. And so at those temperatures, those organisms are active. They are enzymes. They have those specific enzymes that are active at those temperatures. But well, we don't have to worry about those organisms to say, so how about if those organisms, if we get in contact with those organisms, for instance, you don't have to worry about them because our body temperature is 37 degrees Celsius. And the terrestrial temperatures. For instance, you take Ghana, say if you take Accra, our temperatures are mostly around, say 28 degrees Celsius, 27, 26. Maybe when there is heat, you get into 30. Even if you go up north, somewhere in March, February, there about that, the weather can get very warm. It's usually around maybe 35, 37 degrees Celsius, thereabout. So those temperatures are still not optimal temperatures for such bacteria. And so those bacteria cannot be active in these conditions. But you also remember, I think by now you would have done some practical in the lab, or if you haven't, you can note this as some theoretical foundation upon which you'll be doing such a practical. I think you'll be doing some extraction of DNA and do something like a PCR, where you use some thermophilic bacterial enzyme, which you use to, when in the process of the PCR, you heat your nucleic acid mixture in order to break down the hydrogen bonds between the nitrogenous bases of the nucleotides, in order for you to have free nucleotides that can combine in different or various sequences. At the time you're heating the mixture over, say, 60, 70 degrees Celsius, obviously, if you are having an enzyme, like say you are using say a ligase, which will help you to bind these nucleotides to form fragments of DNA. If you do that with an enzyme which cannot withstand such high temperature, the enzyme is going to break down, will get denatured, and so your process will not proceed. And so you use some specific thermophilic bacterial enzymes. So you see, that is a practical application of this knowledge of how temperature affects enzymes. So that even when you heat, say, 60, 70 degrees Celsius, and you lower the temperature again, you still will have those enzymes that will be active in the mixture. All right. Then the last thing is pH. How does pH affect enzyme activity? We see that, say, in our mouth, what's the pH? The pH is about 6 point something, maybe 6.8 and 7.2. So the pH in our mouths is hovering around, neutral pH, slightly acidic, sometimes slightly basic, like that, just hovering around a neutral pH. And so what it means is that the enzyme, which enzyme is active, the common enzyme active in our mouths is what? Salivary amylase. And salivary amylase acts on carbohydrates, breaking down polysaccharides into monosaccharides. And so it means that the salivary amylase, which is a protein, is active at that pH, that optimal pH, hovering around 7, slightly acidic. Again, when you have the pH increasing beyond that, salivary amylase will be inactive. So taking the human body again, if you go into the stomach, the gastric juice is acidic because hydrochloric acid is secreted in the stomach. I mean, this is senior secondary school biology. I think even in junior secondary school, you did some biology of a sort that talks about digestion. And so you can easily relate to this foundational biology. And so what it means is the enzymes which are acting on proteins in the stomach, like pepsin, trypsin, these enzymes will be active at such low pH conditions, acidic conditions. So for pH, it depends on the enzyme and its environment. There are enzymes that are active in acidic medium, and there are enzymes that are active in basic medium. So even though you see that pH is the relationship looks much like that of temperature, here the pH value does not cut across for all enzymes. So a particular enzyme can get denatured at a certain pH, whilst another enzyme will be active at that pH. All right. So these things clear now. I think we actually touched on them, but we have quickly reminded ourselves of these things, and so shouldn't have any problem, with applications of these things. You come to realize maybe if we take implementation of enzymes in, say, the practical application in industry, for instance, then you realize that it is because of these things, what we have just talked about, these factors, that you have, industrial digesters having these parameters being controlled. Let's say if you go to the brewery or you go to some food industry, you see that the factors that are controlled in their bioprocesses are these things: temperature, pressure, temperature, pressure, pH, concentration of the enzyme that they are introducing. So, for instance, if it is beer making and you introduce some bacteria or maybe some yeast, bread making, for instance, take a certain amount of these organisms, introduce them to act on carbohydrates, cause fermentation. So these are the factors that are controlled in the. You see, there's a huge processor there that they have added some feedstock and all of that into it for product to be formed. And on it, yes, you will see these things are connected to a screen or computer where these parameters are being controlled, because you need to have optimal condition for enough yield of the product that is being produced. So those are practical applications. I think along the line, maybe in the next lecture or two, we'll see some of these practical applications. All right. Now let's take mechanisms of action of enzymes. We'll take a look at two major mechanisms. The first is what we refer to as the lock and key model, which is also referred to as the Fisher model. And the second is the induced-fit model, also referred to as the Koshland model. Now, the Fisher model is named after the German scientist Emil Fischer, who first proposed this model of enzyme action. And according to him, an enzyme has what is referred to as the active site. We did talk about the active site as the part of the enzyme that interacts with the substrate. And according to Emil, the active site has a configuration that fits exactly to its substrate. And what it means is that you have, say, a substrate, which is like a keyhole, and the enzyme is the key that fits exactly into that keyhole, the substrate. So that if you have two substrate molecules which are to react, and their reaction is to be catalyzed by an enzyme, what is being depicted below, you have an enzyme molecule that is having two sites. One site fits to the substrate molecule, which is depicted with the round sphere, and you have another site of that enzyme which fits to the second substrate, which has a V groove in it. And these two sites are compatible to each of these substrates, so that you have the round substrate fitting into one of the sites, whilst you have the substrate with a V groove in it fitting into the second site. And then you see there's, say, the reaction is being depicted to have taken place between these two substrates. Let's call it some synthetic reaction, to say that these two substrates have bonded together to form a product. And so you have that enzyme substrate complex, which then changes into, say, an enzyme product complex, and later you have a product which dissociates away from the enzyme, leaving the free enzyme, and then the product which is formed. Now, this model had been used for years, but later on, discussions had shown problems with such a model that talks of an enzyme that fits exactly to a substrate. Now, to understand the problems with such a model, we have here a depiction of, say, a reaction where, let's say, we take an analogy of, say, a metal stick. So a metal stick is supposed to be broken into two. So that would be two products formed out of a metal stick. Take it as it's a molecule which is supposed to be broken down into two molecules, like, say, maybe a hydrolytic reaction, for instance. Now, in the first place, that molecule will reach a transition state where the particular bonds that are to be broken in that substrate will be weak, which is being depicted by this metal stick, which is bent. So when the bond now, according to the Fischer model, so we come down to B. According to the Fischer model, if you take an enzyme, which is depicted with the blue part of this diagram, And because we're talking about it as the substrate as a metal, we take the enzyme to be a magnet, and that magnet is having such a magnetic field which fits exactly to the substrate, so that you have an enzyme substrate complex which is perfectly fit. Now, you do remember we talked about the basic principle of catalytic action in first year. If you have forgotten, you should go back and revise those notes. We did talk about reaction energy profile, and we talk about what we refer to as an activation energy, where substrates that are at a ground state attain activation energy to their transition state before they react and form products. And we talked of the fact that, I use an analogy that compounds, chemical substances will not react to form products if they are in a stable state. So I use an analogy of, say, for instance, as you are listening to this now, whichever position in which you are, if you are comfortable, there will be no point for you to change that position. It is only when you are uncomfortable with a current position that you make efforts to change your position. Same thing at a molecular level. If molecules are at a stable state, there will be no point for them to change into a different form. And so if you are having an enzyme substrate complex that is so fitting, perfectly fit as is shown here, then there will not be any reaction occurring. There will not be any proceeding of a reaction after this, because this enzyme substrate complex is so stable that there will be no need for it to want to change into a product. And so because of that, the modern notion of the lock and key model was changed to say that, in fact, the enzyme and substrate do not perfectly fit, but that if you see what is being depicted in the beginning here, you have the blue part, which is the enzyme, say a magnet, which is binding to the substrate to form an enzyme substrate complex. You notice that substrate is not perfectly fit to the active center, also depicted as, say, the magnetic field of that magnet. So this is not perfectly fit. And because it's not perfectly fit, as the substrate changes its shape to fit, so it means the enzyme causes a certain change in that substrate so that it begins to get fit to its magnetic field, so it's the active site, which is what is being referred to as a transition state. And in that process, that substrate gets deformed. You have a weakening of bonds in the substrate. And so the transition state is unstable. And because that transition state is unstable, the bonds that are in the substrate are also weak. Eventually, those bonds get broken. That metal stick gets broken, and you then have your products and your free enzyme. Now, this brings us to what the modern model of enzyme action, which has been applied over the years, what is being referred to as the induced-fit model, named after Daniel Koshland, an American biochemist. So sometimes this is also called the Koshland model. And referring to the Fisher's model, he said, yes, there's an active site, but that active site does not perfectly fit to the substrate, but rather, in the presence of the substrate induces the enzyme to change its conformation to fit to the substrate. So there's an inducement of that fitness of the substrate. Now, what it means is that if you take, this structure, say this structure with a groove in it, which is depicting the enzyme, and then you have the substrate, which is, the violet shape, which is to get into the active site. If you pay close attention, that substrate does not perfectly fit into that active site. But the moment the substrate gets into the active site, you see the enzyme clamps down on the substrate. So that is the change in conformation of the enzyme to fit to the substrate. We can see it much more clearly, step by step, what Koshland proposed as the induced-fit model. So if you take this, the yellow objects as the enzyme, and the red is a substrate, what is being depicted here is that you have an enzyme, say at the beginning of the reaction, it's having an active site, which is having a round shape. But a part of the substrate, which is supposed to fit into the enzyme, is having a square shape. So this substrate, square, is not perfectly fitting into the active site of the enzyme. But in the presence of the substrate, you notice that there are conformational changes in the active site of the enzyme, so that the active site of the enzyme now changes to have a square shape. And so now the substrate and the enzyme fit, and a reaction on the substrate is catalyzed by the enzyme. Products are formed, which you can see as a breakdown of the substrates into two molecules, released, and a free enzyme is there to carry out more reactions, further reactions. So this was the induced-fit model by Koshland. So these are the two major mechanisms of enzyme action that you refer to. Now, what is the active site? We did talk about the active site as a major, the main or a specific part of the enzyme, the protein, where you have the substrate binding for a reaction on it to be catalyzed by the enzyme. And I've brought, say, a diagram to show it for you to have a better understanding of what we are referring to as a substrate, and also to introduce a new term, what we're referring to as a substrate binding site. What's the difference between the active site and the substrate binding site? Now, let's go back to the fundamental, which is proteins are made up of amino acids. Now, these amino acids are bonded together in peptide bonds to give you, certain sequence of amino acids if we are talking about the primary structure of these proteins. And you also remember that we did talk about secondary structure of proteins, tertiary structure of proteins, quaternary structure of proteins. Now, these proteins, which are acting as enzymes, we usually will be having these secondary and tertiary structures in them. So what it means is that the protein is folded such that you can have specific amino acids that are close to each other. Some 2 amino acids, some 3 amino acids, and these form the pocket-like structure, what is being referred to as the active site, into which a substrate can enter. And so this whole part of, say, a protein, which is shown as the blue color, is the active site. That is a pocket-like structure which is formed by, say, 2 or 3 amino acids that are folded, come close to, like the protein is folded and you have these 2 or 3 amino acids coming close, having some interaction between themselves and forming that pocket-like structure into which the substrate can enter. But even in that pocket-like, structure, which is the active site, there are specific portions, specific part that is going to have interaction with the substrate. So, for instance, if you have an active site, which is made up of, say, 2 amino acids, let's say cysteine or serine, or maybe histidine, maybe these 3 are forming that pocket-like structure. Now, when a substrate comes into that active site, when we say the substrate is interacting with, say, the sulfur hydral group of cysteine, that's a specific binding site we are talking about. Or if it is serine and we say the substrate is having some proton transfer interaction with a hydroxyl group of the serine. So that hydroxyl group of the serine that we are talking about becomes the specific binding site that we are talking about. Now, I will tell you again that when you listen to this lecture and you also read, let's say, a book chapter I gave you some materials even in first year. Yes, you have to go and revise the structures of the amino acids, because I was just talking about, say, serine having hydroxyl group, cysteine having sulfur hydral group. If you have forgotten these things, you must go and look back at the structures of the amino acids in order for you to understand what we are talking about. So you see, in biochemistry, nothing is going to be thrown away. It continues to build up from first year all the way to your last year. So foundation, and we keep building on it. If you forget something, there's nothing wrong. I mean, we all forget things sometimes. You always will just have to go back and refer. Look at the structure of the amino acid again and you have a better understanding of the conversation. All right, let's move on and talk about, say, which amino acids you will usually find in the active site of the enzymes, because we just talked about the fact that the active site is made up of, say, maybe some 2 or 3 amino acids that are in close proximity and they are forming a pocket-like structure that will be having interaction with the substrate. Now you find that in the active sites of most enzymes, there are some amino acids that you come across often, and these are histidine, cysteine, aspartic acid, arginine, glutamic acid, and serine. These are the major amino acids that are found in active sites. And the amino acids that we just mentioned, you ask yourself, what makes these amino acids be the ones that are found in the active sites to be interacting with their substrates? I brought a table below here for you to see the amino acids that we're talking about. I've highlighted them in red for you to see that they have some specific characteristic. For instance, aspartic acid and glutamic acid are having some extra carboxylic acid group in their side chain. And it is that carboxylic acid group that can be involved in what we are referring to as a acid-base reaction, or some acid-base interaction with their substrates. Similarly, you do have, say, arginine and histidine. So arginine and histidine, they are having some extra. Actually, you have arginine having some extra amine group in its side chain. So of course, that amine group will be acting as a base. And you have histidine, which is having what we refer to as an imidazole ring. And that imidazole ring is having a nitrogen in that ring structure. I think we'll see a specific mechanism that you see what we refer to as a proton transfer. So hydrogen ions that go onto that nitrogen. And by that, you have an activation of the enzyme. And you also see serine is having a hydroxyl group. And that hydroxyl group is reactive and can have some hydrogen bonding interactions, or say also proton transfer interaction in the activation of enzymes. Cysteine is having a sulfur hydral group, SH, and so that sulfur hydral group is also reactive, can release protons and have such activation of the enzyme by the transfer of protons from the sulfur hydral group. So I think what we'll do now is we'll take some specific examples of enzyme mechanism. Some can be looking sort of so complex, but like I always say, don't be intimidated by these things. Have patience for yourself and try to make an understanding of what is happening in this, especially if you see reactions that are continuous. Take one arrow at a time and try to see what really is a mechanism, what is occurring, and make some meaning out of it. You may not understand everything all at once, but particularly the first time can be painful. Second time, it will be more meaningful. By the third time, you begin to get it more and more. And as you go ahead, you will understand it. So just have patience for yourself and you'll be okay. So this is an example of an enzyme, what is referred to as papain. I've just brought a diagram, simple diagram. So let's take it that the green is the protein chain. Green is a protein chain. And papain is a big protein. I think papain should be containing some hundreds, maybe close to 1,000 amino acids in it. I don't remember. And okay, King Emahi says he can't see my screen. If you cannot see my screen, I think you're having a network problem. And sometimes I hear the students advising one another that go out and come back in, and then the screen comes back. But in any case, the lecture is being recorded. So when I share the link, you will see all of this. It will show, so you can compensate for what you are not seeing now by watching the video again later after the lecture. All right. So you have this. So the papain, an enzyme which is involved in the breakdown of peptide bonds. And actually, papain is an enzyme which is applied in industry, in the meat industry, in meat tenderizers. So making meat soft. They do that. And even in leather processing also, I think papain is also used there in softening of leather. And we see in the active site of the enzyme papain, there are two amino acids being shown here. One is cysteine. And so this is what I was talking about. Cysteine is having a side chain, CH2SH. That's the sulfur hydride group I was talking about. And you see, there's a number written there, 25. What does it mean? So if you take the sequence of the protein, papain, if you are looking at the primary structure of the protein, papain, and you are counting the amino acids from number 1 to the last, about maybe 900 and something, cysteine, which is at the 25th position, is part of the active site of the enzyme. That's what it means. Similarly, histidine is histidine, which is at the 159th position, is also involved in that active center. Of course, if you take a protein, that protein can contain many cysteine molecules along the chain. Histidine can also be there, but these specific ones, cysteine at the 25th position and histidine at the 159th position. And why is it so? Because you have the protein, its functional conformation, the functional structure is such that when the protein folds, these two amino acids come close. They are in close proximity such that they begin to have an interaction between themselves, so that when a protein enters into that pocket, what we are referring to as an active site, this protein is being depicted here as, say, if you take a protein, the protein is having a peptide bond. And so if this peptide bond is between the carboxylic acid group, the C terminus of one amino acid, and this nitrogen, which is the amine group of a second amino acid. So that N is showing the N terminus. And you see the rest of the amino acid chains are being shown with R and R prime, because we are just interested in the bond that is between carbon and nitrogen. That's a peptide bond, which is supposed to be broken. Now, what you see is that there is some nucleophilic attack by that lone pair of electrons on sulfur on that carbon of the C terminus of one of the amino acids. And obviously, that is going to result into some shift of electrons, resulting into some polarization. And so there's going to be some sort of a stretch in that bond. Similarly, you are seeing that nitrogen in the imidazole ring of histidine is also having some hydrogen bonding interaction, so that nucleophilic attack on the carbon, which is going to be resulting in some polarity there, and the hydrogen bonding with that imidazole ring, these two interactions will result in a stretch and a weakening of that peptide bond, so that the peptide bond eventually can get broken. So that's just one of the, like the active sites we're referring to for the enzyme papping, looking at a specific mechanism of action of the enzyme papping. Can take another one, but before then, let's see some general aspect of the mechanism of enzyme action, which we have an enzyme being shown here, which is having the active site as a pocket, and you have substrate, two substrates, substrate A, substrate B, which are supposed to react, and their reactions should be catalyzed by this enzyme. So let's take a look at a step-by-step process by which that mechanism of action of the enzyme in such a reaction will look like. So if you have the free enzyme with these two substrates which are supposed to react, what happens is you have the substrate, the two substrates, getting into the active site. Bear in mind that we did also talk of the fact that catalyzed reactions that already can occur spontaneously, they only come in to speed up the reaction by lowering the activation energy of the reaction. So you see, you have these two substrates which are interacting in the active site. So the substrates react here in the active site in the third stage, forming a product. And so now you have some enzyme product complex, and then those products get hydrolyzed away from the enzyme. You have a free enzyme and you have your free products. All right, so this is just some pictorial, step-by-step mechanism of action for you to, have a better understanding. Now let's take a more chemical, a chemically presented mechanism of action, and this time we're looking at the action of another enzyme which acts in the breakdown of proteins. This enzyme is chymotrypsin, and it is one of these proteins which act by the mechanism we refer to as the serine protease mechanism. So you have a group of enzymes that have serine, the amino acid serine, in their active site, what I was talking of earlier. And because of the hydroxyl group on the side chain of serine, there is that proton transfer, from serine to another amino acid, which is in the active site, and this results into the activation of the enzyme. So let's see what we are looking at. So like I say all the time, don't get intimidated by this, and you don't have to cramp it in your head. You only have to look at every arrow at a time and try to understand what is happening. That's all. Now we see, so histidine, so this is histidine, it's written there too, so you can see it, histidine. And when I share the slides with you later too, you can also see it well. So you have histidine that is having the imidazole ring, and you see in the imidazole ring there is a nitrogen there. And you see serine, so that is serine, and serine is having a hydroxyl group in its side chain, OH. You see that there. Now, this is the active site of an enzyme. So we're just taking a look at the 2 amino acids which are found in the active site of the enzyme. And this serine, the hydroxyl group, which is on the side chain, is having interaction with that nitrogen in the histidine, the imidazole ring of the histidine. Again, the substrate on which the enzyme should be acting is a protein. It's a peptide bond which is supposed to be broken. And here again, that peptide bond is what is being depicted here, the bond that is between the carbon and the nitrogen. And so you see, the carbon is, say, the C terminus of one of the amino acids in that peptide bond, and that nitrogen is the N terminus of the second amino acid in the peptide bond. And you can see the rest of the chain of the amino acid is just being shown by RR there, because our interest is only on this, on the peptide bond that is between the nitrogen and the carbon. Now, what do you see? In the first place, you are having that hydrogen and the hydrogen of the hydroxyl group of the serine that is interacting with the nitrogen. You know, sort of some intermolecular interaction, which eventually is going to result into a proton transfer from the serine onto that nitrogen. And at the same time, you are having an interaction again on that C terminus of one of the amino acids which are involved in the peptide bond. Because sort of that intermolecular interaction which eventually will be resulting into some formation of a temporary covalent interaction. Now, if you look, the next step, what is happening is that the hydrogen from the serine hydroxyl group has been transferred onto that nitrogen of the imidazole ring. And that hydrogen begins to have an intermolecular interaction with the nitrogen, the N terminus of one of the amino acids. And you have that, temporary covalent bonding of the oxygen of that hydroxyl group, the original hydroxyl group of the serine, with the carbon, the C terminus. May you form this an intermediate. This is an intermediate having such a tetrahedral structure. And eventually, what you notice is that the hydrogen, which had been transferred onto the imidazole ring, is now bonded on the nitrogen, which was the N terminus of one of the amino acids. So clearly, this amino acid has been broken off completely, because you have the NH2 and then there is R over there, the rest of the chain of that amino acid. But you see that the amino acid of the C terminus involved in the peptide bond is covalently bonded onto serine, but of course that is just some temporary covalent interaction there, which eventually you have water in that process. And what is water doing? You see the nitrogen which was transferred from the hydroxyl group of serine, that hydrogen has to be gotten back from where? Water. Water now brings, you have water which gets ionized into hydroxyl and hydrogen ions. And so you have that proton which is transferred from water back onto the nitrogen of that imidazoles ring. And in that process, you also will have again that nucleophilic attack, say the hydroxyl group of the water will now be having such an attack on that covalent interaction which is occurring between the serine group and the C terminus. Eventually, the proton is transferred onto the nitrogen, and you also see that you have the hydroxyl group binding onto that carbon, which is the C terminus of the second amino acid. In the last end, you see the hydrogen which was donated by water, which was transferred from water onto that nitrogen. That hydrogen is given back to the serine residue. Now, what do you notice? You also notice that the second amino acid is also now released from the serine residue. And the hydroxyl group from water has now fully bonded onto that carboxyl group. And so you now have also a full amino acid. And you also have the active site regenerated, because the original active site should have serine having that hydroxyl group on it and have that imidazoles ring. And it is that original active site that will then have that hydrogen from the serine interacting with the imidazoles ring for the transfer of that hydrogen ion, or what you refer to as a proton, being transferred onto the nitrogen, activating the enzyme again, and it can carry out another reaction. So this is one of these mechanisms that you find proteins, enzymes like chymotrypsin undertaking to break down proteins. So you see, usually we do talk about, say, enzyme breaking down, say, proteins. And when you say an enzyme is breaking down a protein, remember that enzyme itself is a protein. So you see the chymotrypsin is a protein, and it's having some specific amino acids at a particular position, some particular positions of its structure interacting on the peptide bond of another protein, which is supposed to be broken down. All right, so you have to listen to it again and again. You have to watch this. I understand that sometimes some of these things, the first time you may not clearly understand it, but you have to make the effort. Look at it a second time. Look at it a third time. Somebody will understand it a second time completely. Somebody will understand it right now, first time. Somebody the third time. Somebody the fourth time. Somebody the fifth time. That's why I do say have patience for yourself and everything will be okay. Okay, I think maybe we should take maybe a last one, another mechanism of action, and then we can open the floor for any questions, comments, The chart is working. I'm monitoring it. So if anything, you can write there. We take in another enzyme, lysozyme. And the lysozyme is a hydrolase. What are hydrolases? Enzymes that catalyze hydrolytic reactions. And this is an enzyme which is applied in antimicrobial substances like some bactericidal substances. And what do? Lysozyme breaks down what we refer to as the cell walls, the peptidoglycan cell walls of bacteria. The cell walls of bacteria are made up mainly of this compound, this polymer, what we're referring to as a peptidoglycan. And that peptidoglycan consists of these derivatives of monosaccharides, which also, I think, we looked at it when we were looking at carbohydrates briefly. We may have, this is HND, so you would have done it in SCT 106, first year. N-acetylglucosamine, that's a derivative of glucose, and N-acetylmoramic acid. So these two, N-acetylglucosamine, N-acetylmoramic acid, form a polysaccharide. So you have these glycosidic bonds between them, and you see these monosaccharide derivatives are alternating. So you see N-acetylglucosamine bonded to N-acetylmoramic acid, and next is another N-acetylglucosamine bonded to N-acetylmoramic acid, and so the chain continues that way. And along the and then also within this chain you have some peptides which form some cross linkages, which is why that is being referred to as a peptidoglycan. The peptidoglycan simply is telling you that there is some peptide and you also have carbohydrates that is giving you the full structure of that particular substance. And it is this bond, let's say this glycosidic bond, is to be broken between the N-acetylmoramic acid and the N-acetylglucosamine. This bond is to be broken, and that bond will be broken by hydrolytic action. And that hydrolysis, that process, is catalyzed by a lysozyme. And so you see, if you have the peptidoglycan wall, the peptidoglycan being the major component of the cell walls of bacteria, and lysozyme can break down the peptidoglycan, then of course what is going to happen is that it will result into the bacterial cell death, because once the cell wall is broken, it's like the bacteria ruptures, its contents release, no more cell, and that's it. Bacteria is destroyed. So that's the mechanism of action of the lysozyme there. You can see it more clearly here, where you have in the lysozyme active site, you have aspartic acid and glutamic acid. And so that's aspartic acid and glutamic acid will be having some interaction with this hydroxyl group, these hydroxyl groups, and even the hydroxyl groups of the acetyl group of the N-acetylglucosamine in the active site. So the active site, you all remember the aspartic acid, glutamic acid. We did mention the fact that in the side chain of these amino acids, you have a carboxylic acid group there. So those carboxylic acid group can have such hydrogen bond interaction with the hydroxyl group, which is on what you see is say carbon number 6 of that glucose derivative. Or if you look at it clearly, this rather should be the carbon number 6. And so if you count the carbons, then of course this is going to be like, say, carbon number 1, 2, 3. Like on the carbon number 3, you have a hydroxyl group which is having such an interaction with a carboxylic acid group. And then the N-acetylglucosamine, which is the acetic group there, is also having some hydroxyl group there that can also have such an interaction. And here you see, or just showing that there's a glycosidic bond between the N-acetylmuramic acid and N-acetylglucosamine, which is being hydrolyzed, and that hydrolytic reaction is being catalyzed by a lysozyme to break that bond, so that you now have N-acetylglucosamine, N-acetylmuramic acid, and N-acetylglucosamine separating. So the bond is broken. And if that is the long chain there, if that bond is broken there, then of course the cell wall is broken. So the bacteria will have no longer protection. That will result in cell death of that bacteria. I mean, this is also one of the applications which you find in the pharmaceutical industry. So I think today we've looked at this application in pharmaceutical industry. We talk about papain, which I said it can be used in leather industry. It can be used in the meat industry as well. Chymotrypsin is one of the enzymes also used in the food industry. Also it's involved in the digestion of proteins. All right, so this is where I will open the floor, and if you have any questions, you have any contributions, you can raise your hand. I will enable your microphone, call your name, you speak, or you can write in a chat. The floor is open.